This lecture is going to go briefly over genotypic methods of diagnosing infectious disease. It's going to include molecular methods, so PCR, whole genome sequencing, as well as pulse field gel electrophoresis. Those are the ones I'm going to focus on. Your text goes into a few more, but these are most commonly used in clinical labs and public health labs to diagnose infection as well as identify microbes that could be part of an outbreak. Polymerase chain reaction, or PCR, is a technology that is employed across a lot of different fields, microbiology being one of them. So anytime that there's a DNA sample, PCR can be used to amplify that section of DNA to many magnitudes in numbers and copies. And then those copies can either be ran out on a gel, and you can look at their banding pattern, or they can be analyzed by fluorescence and watching the numbers of copies increase through what's called real-time PCR or quantitative PCR. This is widely used for viral work to identify if certain viruses are causing an infectious disease by amplifying genes that are specific for that virus. So COVID testing, you've probably heard of the PCR test for COVID. What that's doing is it is amplifying a set of genes that are unique to the SARS-CoV-2 virus. And if those genes are present, they get amplified. You can view that in a real-time manner or a quantitative manner, and then that patient would have that virus. Another type of PCR also has like an RT in front of it, but it's reverse-transcriptase PCR. And with reverse-transcriptase PCR, that is actually creating DNA out of RNA and then amplifying the DNA. This is used for RNA viruses like SARS-CoV, as well as influenza viruses and RNA virus as well. The deal is that with PCR, it's going to amplify DNA. And so if you're pulling genes from an RNA virus, you have to first convert them to DNA to make copies of it. But anyway, the process is the same. It's DNA amplification, looking for the presence of a certain set of genes, which would indicate that a pathogen is present. Whole genome sequencing, or WGS, is a process of taking the genes of a microorganism and sequencing all of them within a cell. This is done more and more frequently, and we see whole genome sequencing most employed when we're looking for the connection of an outbreak. For example, if we have an E. coli outbreak, any patients that test positive for E. coli would have those cells sequenced, their entire genome sequenced, and you would look for matches across patients, because if two different patients have E. coli with the same genome sequence, that must mean that those two E. coli samples were derived from the same original E. coli, and so you can connect people in an outbreak. It used to be very expensive to do whole genome sequencing, but the cost has really come down, the ease has really come down, and more labs, even small labs, are starting to do more sequencing methods. And therefore, whole genome sequencing is a pretty common practice now in public health labs and larger clinical labs as well. An alternative to whole genome sequencing is pulse field gel electrophoresis. In this process, the DNA is still being analyzed from a particular cell, but rather than taking and running the DNA through a gel in one direction, the DNA is actually broken up into lots of different pieces. and then electricity is applied in different directions across that gel to allow even a more separation of these DNA fragments. And so instead of having bands of certain sizes, we have movement of different pieces all over the gel. But what's really awesome is that bacteria that are clones of each other, they're all going to move in that same path. And so you end up with a DNA fingerprint. Essentially, every strain of bacteria will have their own pattern. And then those patterns can be compared, where if you have a similar pattern or a similar DNA fingerprint, then we can connect those species or those strains, those patients together. And so it's definitely used in watching an outbreak. And the CDC uses pulse field gel electrophoresis for, you can still do whole genome sequencing, but this is becoming more and more common method. As I mentioned in the previous slide, the CDC really uses these microbial fingerprints produced from pulse field gel electrophoresis to follow an outbreak and connect individuals. And all of this information is stored in this giant database called PulseNet. And so public health units across the country all have access to this PulseNet. And when pulse field gel electrophoresis is done, then these results are uploaded to PulseNet, and then CDC researchers epidemiologists can follow these fingerprints to try to pinpoint an outbreak source. And this has really cut down on the time it takes to identify a source from previous methods of data analysis and data acquisition.